Okay, so let's get started. Welcome to the first HEAS seminar in Ancient Genomics of this round. We're very pleased to have Robert Kofler here. He is an expert in population genetics at the VetMed University here in Vienna. So he's a local guy. Actually, he comes from agriculture and plant breeding, where he did his degree and PhD at the BOKU in Vienna here. And yes, so as I said, now he is a faculty member at the VetMed and working on insect evolution and other fields of genomics. And he will tell us today about museomics of, well, Drosophila and insects.
Thanks, Martin. And thanks for inviting me and giving me the opportunity to talk here. Yes, let's go on. Okay, my group is interested in the evolution of genomes. I mean, you all know genomes is the Bauplan to generate us humans. And yeah, you need a model organism for this. And one excellent model organism is Drosophila melanogaster. So why is it such an excellent model organism? It has a short generation time, a small, well-characterized genome. This is especially important if you, don't want to spend too much money sequencing the genomes, like humans, way too expensive. Many publicly available resources exist, many genetic tools, and it was the organism of choice by the founding fathers of genetics. This is a picture of the founding fathers with Thomas Morgan, Calvin Bridges, and Alfred Sturtevant. And let's remember Alfred Sturtevant for later. Okay, and recently a unique opportunity arose to study how genomes are actually evolving. People published the genomes of 20 Drosophilas from the museum. So there are 200-year-old Drosophilas in the museum in some drawer. No one cared about them. And then people took them, dipped them for 2 hours in a solution, in some DNA extraction solution, dried them and put them back into the museum drawer. So it was non-destructive, minimally harmful. And the DNA in the solution was sufficient to sequence the entire genomes. This is some pictures of the specimens. There are also pictures before and after. The procedure had little effect on the specimen. Maybe the eye color was a little bit, lost a little bit of the brightness, but in general, it had little effect on them. So it's a nice way to sequence the genome of this historical specimen without doing any major harm to them. And we were interested and we thought, okay, let's see what changed in this genome since 200 years and now. So what we did is we took the reads and aligned them to the reference genome, then we noticed one very weird thing. So there are reads. These are reads from the museum. I guess you all know how an IGV screenshot looks like. So these are all reads aligned to the reference genome. And what we saw is, okay, there are tons of reads aligning here, and then, damn, there is a gap. There are no reads aligning here. And then many reads are aligning again. And this is very strange. How is this possible? What does this mean? This means actually something is missing in the genome of the 200-year-old flies that is present in the modern flies. So there is a puzzle piece is missing. And then we checked, okay, where are these missing puzzle pieces? They are everywhere in the genome of Drosophila. It has like a couple of chromosomes, 2L, 2R, 3L, 3R, X. And when we checked where are these missing puzzle pieces, they are here in chromosome 2L, 2R, 3L, 3R, and X. They are basically everywhere distributed in the chromosomes. We extracted the sequences of these things and clustered them. You can just check which sequences are similar and realized that they belong to 3 big groups, the yellow group, the green group, and the black group. And again, these groups are distributed everywhere in the genome. Okay, so we just blasted it and realized, it's transposable elements. There is, the yellow group is actually blood, it's called blood. The green group is opus, and the black group is 412. Okay, what are transposable elements? It's like the viruses of the genome. It's selfish DNA that is jumping in genome, multiplying, and making copies of itself. Yes. And why are transposable elements actually important? Well, there are major drivers of phenotypic evolution. One very nice example is tails. You may have noticed we don't have a tail anymore. We lost our tails. And the main reason is an ALU-Y insertion in this hominoid TBXT gene, whatever. This is so a transposable element insertion is actually responsible for us having lost tail. Another beautiful example is the peppered mouse. The peppered mouse. So we all know that this is how the peppered mouse would look like, you know, very whitish. But then industrial revolution came, the trees turned from, I think it was the birches, turned from whitish to blackish, and then these guys were eaten by birds. So the color, this is called industrial melanism, changed from this whitish to blackish of the peppered mouse, and they were less eaten by birds, and so they've survived. And the thing that is also responsible for this very rapid adaptation, and it was a rapid adaptation, is again a transposable element insertion. It was published 2016 in Nature. Very nice publication. Okay, but the funny thing now is that it's actually turning back. It's reverting. So the peppered moths are now more looking like this again because, you know, the industrial pollution decreased, and now peppered moths look more increasingly look like this again. So it's a very nice example. One question that you may have is, okay, where are they coming from? Where are these transposons coming from? They are largely vertically inherited, so from parent to offspring, but occasionally they are horizontally transmitted. So what does horizontal transmission mean? For example, if a transposon is jumping from the rat to the horse, that's just a stupid example. This is a horizontal transmission. And horizontal transfer was actually discovered by Margaret Kidwell. She did amazing work in this field in Drosophila, and it all started with a weird phenotype. She realized if I cross flies from this white line to the red line, I mean, they're not really red, this is just, you know, me showing that there are differences, then you get, these are ovaries of Drosophila, they are healthy, wonderful, this is how ovaries should look like. But if you cross a male from the red line to a female of the white line, this is the ovaries you get. They are totally destroyed. They are unhealthy. And now the important thing is, these guys and these guys have totally the same genome. But here, the ovaries are destroyed. Here, the ovaries are healthy. This is called hybrid dysgenesis. And later, people realized the only difference is that the red lines have a transposable element that the white line doesn't have. It's called the P element. And this is the reason why the ovaries are so destroyed. And yeah. Okay. And one further mystery was discovered that all the nines that you capture in wild population are the red ones. It means they have the P element. Only some very old lab strains that are collected in the 1920s, 1980s, 1930s, and have been maintained in the lab for 80 years and so on, they are the white ones. They do not have the P element. So whatever, if you go out in nature, you sample one fly in Europe, bang, it always has the P element. You can only have some non-P element strains, the very old ones in the lab. It was realized later that the P element was spreading between 1950 and 1980 in the worldwide populations of Drosophila melanogaster. It's coming, there was a horizontal transfer, probably in South America. So the P element jumped from Drosophila willistoni from South America into melanogaster. And then it spread in all populations, and now it's actually found everywhere. So the P element is everywhere, and it spread within 30 years in all populations. So this was a rapid invasion of this transposable element. Back to our transposons, blood, opus 412. Could this actually be also due to recent invasions? Yes. So we see that all these three transposons are absent in samples from the 18th, 19th century, but present nowadays in like in samples from 1933. So if the copy number of 412, 0, and in 1933, not 0, 50. Blood, very low copy number, bang, 20. Same for opus. And if you just show like some other ones, Circe and Invader, copy numbers didn't change a lot. So only these three guys were absent in the 19th century, but present in the around 1933. Okay, and we also were able to figure out where they're coming from, where this blood, opus, and 412 coming from. They are coming from a related species, Drosophila simulans. So if you extract all the sequences of those transposons, make a phylogenetic tree, you see that all the sequences from Melanogaster red are nested within the simulans, Mauritiana, Seychelles. These are related species from the simulans complex. So the donor, there was a horizontal transfer, very likely from simulans, perhaps this guy, to Melanogaster, where it was spreading in the 19th century. Okay, but now we got really suspicious. So we, like Kidwell found an invasion, we found a lot of invasions in the 19th century. Could there be even more? Could there be even more invasions? I mean, the problem is that all the transposon invasions that we were found were based on the reference genome and the reference annotation of transposable elements. A novel transposable element that would not be present in the reference genome would have been missed. So we were looking for an approach that finds transposable elements without transposable element invasions without having to rely on the reference genome. And I illustrate this with the Habsburg. I'm Austrian, so I have a thing for Habsburg. Let's see. Okay, let's imagine that these are the old Habsburgs and these are the new Habsburgs. And we have a long read, nice reference assembly for one of these new Habsburgs and perhaps old museum data for the old Habsburgs. And now we have a transposable element invasion in the middle Habsburgs. What we would see is if we align reads from the old Habsburgs to the long read assembly of the recently collected Habsburgs, we would get gaps, like I showed before. We would get gaps in the alignment. This means there are reads from the old Habsburgs aligned to the reference assembly of the new Habsburgs, and the reads are everywhere except for those sites where a novel transposable element invaded. And we turned this into a software that we call Genome Delta. So basically the thing is extracting the sequences of these things where no reads aligns, is clustering them, creating a consensus and doing some quality checks and so on and so on. And with this, we have a software that works for model and non-model organisms without having to rely on a repeat library, because repeat libraries are the major pain in the behind. They are terrible. They take hours to generate. Philip, how long does one Lepidoptera take? 2 days, 3 days, 4 days? So it's a terrible effort, and it works more or less. Sometimes it works, sometimes no. It's a pain. And if you can avoid that, that would be fantastic. And with this software, we have now a way to avoid it. And this means we can identify transposable element invasions in any species because we don't need this reference library. I mean, but we still need a reference genome. That is important. Okay. Yeah, so we applied this software to recently collected strains. So there are some people who made like long read assemblies of Drosophila from Europe, they collected the Drosophila in 2021, made a long read assembly, and we used this software and we found, okay, there are several more gaps. Mekele, Suslik, Transip, and so on. And when we now, Drosophila, we are lucky. We have resources, tons of resources. We have 1,000 strains collected during the last centuries. So here is a timeline. There are some strains collected in 1815, 1850, 1925, and so on. This means we have a very nice time series of genomes available that me, as a bioinformatician, can just parasitize. I don't have to generate them. They're available. You're just downloading them and using them. And here on the y-axis is the copy number of the different transposable elements. Red, of course, means it's present, and green means nothing, all chill. And we start with the P element. I hope you remember, it's this transposon that Margaret Kidwell discovered that was invading between 1950 and 1980. So P element as a reference, yes, absent, 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 bang, present. Then the new invaders that we found, Spoink, Mekele, Suslik, Transip, and Transip even 2015. So it's a very recent invasion, just 10 years ago. So now finally we have, for the very first eukaryote model organism, or any organism, I think, the complete invasion history of transposable elements. When to summarize this, 412 blood opus was invading somewhere between 1850 and 19. Then Tarrant was coming in between 1925 and 50. Then I element, Hobo, P element, Spoink, Mekele, Suslik, and Transip. Like in 11 invasions in 200 years of transposons spreading in the genome. Cannot be healthy for the poor flies, no? And but now more complete because we just found another one, Verruca, that is just spreading now. It is present in North America and Africa and China, but not in Europe. But I guess it will come to Europe very soon. Anyhow, but for these two, we are super lucky because there are consortia like the Roseu that are collecting flies all over Europe and the world each year. There are many people that are collecting Drosophila each year from different locations around the globe. This is a perfect opportunity for us to see how these transposons are actually spreading geographically. Let's start with transit. 2014, all green, nothing, no transit. 2014, we see here some red spots emerging here, here, and here. 2015, it's half red, half green. 2016, everything red. So this means in a mere 2 years, this transposon was spreading in all populations, which is crazy fast. I never expected this. I mean, I guess no one did expect this. And there's a nice comparison, Verruca, much more chill, much more slow. 2010, nowhere to be found. Before 2010. Around 2010, it pops up in Africa, which makes sense because we realized that the donor is Drosophila erecta. This is occurring, Drosophila erecta has a habitat here in Africa, so it makes perfect sense. And Verruca basically is the Swahili word for jump. So this is why we chose the name Verruca. And then 2017, it's everywhere in China and North America. 2021, yeah, it's like spreading more, but you see it's more slow. And we checked recently in Europe, it's still absent in Europe. So that may actually be a very nice opportunity for the community to study how a retrovirus, because Verruca is a retrovirus, is spreading in natural populations, and maybe even how the host defense against these things pop up or is established in natural populations. So I'm wondering if this would not be a fantastic opportunity for the community. But yeah, I need to convince the community. Okay, like to summarize, there was a lot of work for us to generate, for us and others, so with Kidwell, Pascal, blah, blah, lots of previous groups established found these transposable element invasions, and our group recently added a couple of novel transposons. So it was a major effort, but now we have the complete invasion history for one model organism, for one of the major model organisms in genetics, actually. But this raises the question, is such a high rate of transposable element invasions normal? I like this comparison because I was at the talk from, I had a talk in China recently from, how is this guy called, who is working with this giant lungfish genome? Axel. Shit. Anyhow, so he was super proud. He was super proud. The genome of the giant lungfish was growing by 300 megabase pairs per million years and reached this gigantic genome size. And then I was wondering, okay, but how much was the Drosophila genome growing in the last 200 years? And if you extrapolate this to 1 million years, then it was actually growing by 5,000 million base pairs. I mean, these 11 invasions, these 12 invasions, increased the genome size by 1 million base pairs in 200 years. This is an enormous rate of evolution. If you extend this to 1 million years, then you would get this. But if you compare the genome size, it's not larger than the giant lungfish. It's actually much smaller. How can we explain this? How can you explain this, that you have a much faster growth in genome, but a much smaller genome? I mean, there are many explanations. One, several people suggested, or maybe it's also shrinking at the same time, at a similar rate. But I don't believe that. I think the more likely explanation is that the rate of invasions that we see in the last 200 years is not normal. I think it increased recently. And how could it increase? I mean, let's pick the number one culprit for everything, human. So there are things, how could humans be responsible for this? Like, we can biological pest control with baculoviruses. I explain these things later. Global warming increases the rate of activity, or habitat expansion of insects. And I have some examples for all of those things. baculoviruses. They are used as biological insecticides. And then people have this, this is from a company. You have Litovir, Helicovex, blah, blah. For the cabbage loper, you have Lupex. Different baculoviruses, how these pests are controlled and biological. So people are very proud, oh, we are not using insecticides, we are using biological insecticides, baculoviruses. But the problem is that there is a very nice study that showed that baculoviruses may be the number one vector for horizontal transfer. I mean, let's be clear, we don't know what is transferring transposons horizontally, could be anything. But if there is one good candidate, then it's baculoviruses. So we may actually end up by fighting them biologically by contaminating the genomes of many Lepidoptera in non-reversibly with transposable elements. We may actually trigger an enormous wave of horizontal transfer in these Lepidoptera by fighting them with baculoviruses. That may be a very terrible idea, and it would be good to follow that up. Come to this later. Okay, baculoviruses, number one. Another thing is global warming may increase the success rate of transposon invasions. I mean, one thing that many people don't realize is that most transposable element invasions fail. I mean, I like to simulate things as well. I'm not just working with ancient DNA. I like to simulate transposon invasion. And this is my simulation of a fly population. So every dot here is a fly. It's a bit abstract. And then I introduce the transposon in one fly. And if you wait for 3 generations, the transposon is lost. And okay, then I introduced it again, and I waited for 10 generations, the transposon is lost by genetic tree. And in 820 out of 1,000 invasions, they failed. So like what we see can only be the tip of the iceberg. There must be much more horizontal transfer. Most invasions will fail, but the success rate depends on the activity of the transposon. If some transposon is making a lot of copies of itself, if it's highly active, like a highly active virus, it will have a much higher success rate. If a transposon is like Karuka, not spreading, very chill, not spreading at a low rate, then the success rate will be very low. But the funny thing is the activity of many transposons depends on the temperature, like the P element, super active at 29, not doing anything at 21 degrees. So actually global warming could not have any effect on the rate of horizontal transfer, but on the rate of how often these transposons are established in a population. And then the last thing, and probably the best candidate, is habitat expansion. One very nice example is that, for example, 3 of these invasions were triggered from South American species. But the problem is, 3 of the invasions in Drosophila melanogaster come from South America. But the problem is, this is how the habitat of Drosophila melanogaster looked 200 years ago. You don't see it in South America. It was happy in Africa and Eurasia, but not in South America. But only in the last 150 years, Drosophila melanogaster extended its habitat into Drosophila willistone, into the habitat of Drosophila willistone, which is here. And this enables the horizontal transfer because these transposons are coming from Drosophila willistone. So these habitat expansions may actually be one of the number one reasons why we have like at least 3 invasions. The other 9 I cannot explain with this. But at least 3, it's a start. So to summarize this, humans may drive increased rates of transposable element invasions. So you can either increase the rate of horizontal transfer, for example, with baculoviruses or habitat expansion, or you can increase the success rate of invasions, for example, with global warming. Okay, but what about other species? Well, Drosophila simulans, closely related to melanogaster, we found a similar thing. These transposon shelters, Boeing, were absent in 1960, but then present in 1990. The P element, the one that Kidwell found, also jumped to simulans around between 2000 and 2010. So we find it in related species as well. Then we find it in Drosophila mauritiana. I mean, for Drosophila mauritiana, we don't have a great time series data. It's like an island endemic somewhere in Mauritius. So we don't have a lot of Drosophila samples from there. Also, I found many volunteers who would like to collect flies from Mauritius. that would not be a problem. And similarly, in Drosophila seychellia, we found even a very weird thing that the transposons are present in some islands, but not on the other. And we think that this must also be like an invasion that is just limited at the moment to these islands and has not spread to the other islands at the moment. So we see it in Mauritiana, Seychellia, and just shelter also in Drosophila thaiseri, where it is present in this population, but not yet in the others. And we observed one thing that is, I find it very cool and but also a bit troubling, a thing that we call cascades of transposable element invasion. So for example, if Drosophila melanogaster, if a transposon jumps from Willistone, which has a habitat here, to melanogaster, which has a global habitat, and then we think melanogaster transmitted it to the island endemic, Drosophila seychellia, Drosophila mauritiana, and Drosophila thaiseri. Because I mean, shelter and spoing, these guys that are invading here, you know, in Mauritius and the Seychellia, they are coming from Willistone, from South America. How the hell is a transposon from South America coming to the Seychelles? You know, did it take a flight or whatever? It can only do so mediated by an intermediate host, melanogaster. So it jumped from Willistone to melanogaster, and then melanogaster distributed it to all these island endemic ones. So even if you are not expanding your habitat like Seychellia or Mauritiana, you're just sticking in your island, not doing any harm to anyone, you may still get contaminated with transposable elements that the global population transmits. So yes. Okay, to summarize this now very briefly, we have 12 invasions in the last 200 years. It's a lot. They were also found in related species, and these recent invasions reshaped the genomes dramatically, increased the genome size by 1 million base pairs. One important question is, what else are they doing? Just increasing the genome size, while they're also having other effects? Many reports show that they are making a ton of recessive lethals, like just the P element. 10% of the insertions are estimated to be recessive lethals, cannot be good for this organism. And we also now got contacted from some researchers in the US. They found that Transib and Koruka, the last two invaders, are creating a ton of recessive lethals. So this must be an enormous load on the health of the population, of the fly population. This must be quite a burden. And they create inversions, translocations, and so on and so on. So they are messing up the genome quite dramatically. But anyhow, this would be one further line of research that I'm quite interested in. What the hell are these things actually doing? And then recent invasions are probably very likely triggered by humans. So the question is, are we entering an area of anthropogenic genome change? Like one of my first attempts to make graphics with artificial intelligence. Good news, it worked. But now I need to pay. Anyhow, so, but we need to test if humans triggered widespread genome invasions in many different insects, and everything from here on will be huge collaboration with Ron and for people from his group like Olivia. Okay, so the first thing we did is we went to the Smithsonian and checked if we can get old genomes from different fruit flies. So the Smithsonian in Washington, and the funny thing is they had the fruit flies, the Drosophila simulans, that were discovered by Alfred Sturtevant. So this is the oldest you can get. He described Drosophila simulans in 1919, but the specimens are from 1915. So we sequenced those flies from the 1915s, and we have some first results, and probably I shouldn't say it online. Between 1950 and 1960. It seems very promising. So this seems to have worked quite well. And but it's largely a work from Sarah and Prakash, and we are currently working on the publication. But let's see. Then I would be very curious to see how widespread this is in many different Drosophila species. Is this just something you see in Melanogaster and simulans? Some people even argued this is exactly the reason why Melanogaster is the model organism, because you have so many mutations. You know, there are the famous mutations where Sturtevant and Morgan established genetics like the white eye. And white eye is a transposon insertion. It's estimated that many of the phenotypic mutations you see that were used to establish the field of genetics are actually transposon insertions. And that's why it was chosen as a model organism for genetics. But let's see. But it will be I think it will be super important to see how widespread this is, how many Drosophila or other fruit fly genomes have that. So we are initializing this, what we call the 1001 historical Drosophilidae genome project. And it's a collaboration with many museums. So I contacted a ton of curators, 10% responded. But anyhow, in the end, we got several that are super interested in this. And I think once the project gets going, I think even more may become interested, maybe even 20%. So Senckenberg, then we have Florida, Leibniz Institute in Bonn. Of course, bless God, the Natural History Museum in Vienna, they're quite interested in this, Oxford Museum, and the Smithsonian. And yeah, this is just an overview of some of the species that would be particularly interested to us. And so the aim is to make a complete invasion history, like I've shown before for Melanogaster, for around 30 different Drosophila species. Because then, when we have the complete invasion history for many Drosophila species, we may be able to identify. So first of all, we will see, is this common to all of them? Can we identify risk factors, like habitat expansion or maybe habitat in South America that is responsible for this? So what is responsible for this? Are there any commonalities in the species that have a high rate of invasion? So that would be this. And then another project that we have not yet triggered, but we would like to trigger in the future, is to see if biological pest control contaminates the genomes of Lepidoptera non-reversibly, like baculoviruses are used for example, Trichoblusioni. And the question is the genome of this Trichoblusioni contaminated by biological pest control? But also, are there off targets? Are the baculovirus also contaminating the genomes of, for example, iconic specimen like the Wiener Nachtfeuernauge? And we have some first results. You cannot do DNA extraction with the entire Lepidoptera. In this solution, if you dip this Lepidoptera into the solution, I think it may end up quite naked. So we used only a leg. We used a leg and did DNA extraction and sequencing, and it seems to work very well. We got quite a decent rate of endogenous reads, 30 to 60%. We are quite happy with that. And this is a project we want to develop soon. Then we have one going with Platella germanica. It has some similarities. It's the German cockroach. It has some similarities to Drosophila melanogaster. It has an origin here in Asia and then recently extended its habitat 200, 300 years ago into Europe and then 120 years ago into North America. So it may be a prime candidate if habitat expansions are actually triggering this. Then we should see it in Platella germanica. Only problem, it's making me very poor. It has a genome size of 2 gigabases, almost like humans. This stupid cockroach is almost same genome size as humans, so it's a very expensive project. And anyhow, the sacrifices I have to bring. So what we are doing here is we have 175-year-old Platella germanica from museums. And we also compared to recently collected ones. Sarah, my PhD student, collected some recently. I should probably not mention where we collected them, but if you ever want to, have nice food in Vienna, maybe reconsider your choices. And then, of course, it will be nice to move beyond museums. In a museum, you can only get 200 years. And these are some ideas that we had. To be honest, it was actually some ideas that Ron had. It's largely Ron's ideas from Inca headlouse, which is 500 years old, the Littoria snails, which are Philippus is right, 10,000 years old. And this The Inca head loss seems to have worked quite well. We have 10 to 20-fold coverage, if I'm not mistaken. Littoria snails, we have first trial sequencing. I think this was also quite promising, 10 to 30% endogenous read. And they are 10,000 years old. That's very nice. And Mus musculus, I think, if I'm not mistaken, this also worked quite well. We got, but let's see. And then there are some even more crazy ideas. And yes, we have to try Jurassic Park. But we're not doing it with million-year-olds. The idea is to try copals that are just 10,000 years old, which is nice because they are, first of all, cheap. You can easily buy them. And yeah, second of all, the DNA may not be so degraded. But we have not yet tried it. But Philip has bought some and could be very interesting. Then one super cool idea was to use Diptera pupae from Avar graves that are 1,200 years old. But we have first results from this, and this doesn't look so promising. I think we have 0% endogenous read. But let's see if we can still do something about this. But at the moment, it doesn't look overly promising. And the one other crazy idea is lake drilling cores. We are collaborating with University of Basel, Margarita Schäfer and Anton. And they have like insect remains from this drilling, from the lake drilling cores that are about 5,000 years old. They will send them to us soon. Let's see if this works. It would, of course, be fantastic if this works. And yeah, okay, acknowledgements. I'm almost done. So Ricardo and Almo did most of the work. Ricardo, Almo, the one that is published. And Anna, Philip, and Sarah are currently working on the museum data. Yes. And then special thanks to the wonderful collaborators, like especially Ron and Olivia and Jared, and also Sarah Senior from the VetMed, from the museums like Smithsonian, especially also, of course, the Natural History Museum of Vienna, where we, Martin, Elisabeth, Susanne, Alexandro, who is the curator from the Diptera, Susanne is from Platella, Sabine is Lepidoptera, and Carlos are also Lepidoptera. And from the Gregor Mendel Institute, they are working with plants, but they actually had this idea with the Lepidoptera that baculoviruses may end up contaminating them. So Anna, Igor, Klina, and perhaps Magnus will also collaborate on this idea. Yeah, Ferran Antolin, and Margarita, and Matthias, and yes, Gabriele Jara with the pupae from the Avars. Yes, okay, many thanks for your attention.